organs that are accessory to the digestive tract. Now, Dr. Banks, I think, either did or is going to give you a lecture on the embryologic development of the digestive tract. Have you had that yet? Okay, so you remember that the pancreas and the liver both grow from the primitive gut. Remember that? So that's why we're studying them with the digestive tract. They're accessory to digestion. All right, so the pancreas, we'll start with that, and then we'll go to the liver. So the pancreas lies against the posterior abdominal walls. It is retroperitoneal. I think Dr. Escobar explained that term to you. It has an uncinate process, and let me show you this little hook. The word uncinate means hook-like. So this is the uncinate process. These blood vessels are the superior mesenteric artery and vein. And notice that most of the pancreas is anterior to that artery and vein, but the uncinate process actually goes posterior to the superior mesenteric. So it hooks around the superior mesenteric vessels. Then we have the head, which is this part here, the body, which is this main part here, and the tail, which, as Dr. Escobar says, likes to tickle the spleen over there. So I don't know if she said this to you, but she always liked to say that the spleen is sort of a two-timing organ. It's cuddling with the duodenum on one end and then playing footsie with the spleen on the other end. So, all right, so the pancreas, like many other organs in the body, is about 25 centimeters long. It's about 5 centimeters wide, 1 to 2 centimeters thick. It weighs about 150 grams. Do not memorize those numbers. I will not test you over them. Subdivided into lobules. We've seen many organs, many glands that have a capsule around the outside, and the capsule extends in and forms little septa that break up the organ into lobules. And so the pancreas is a good example of that. The septa contain nerves and blood vessels. Now the pancreas has both endocrine and exocrine function. It's a very important organ for digestion, but also for carbohydrate metabolism, as you probably already are aware. Okay, let's talk about the exocrine part of the organ first, and then we'll look at the endocrine function later. It produces about 1,200 millimeters of fluid per day. This fluid contains digestive enzymes, and we call this pancreatic juice. Nice scientific name. I would not want to drink a glass of pancreatic juice. I don't think it tastes good. The functional unit of the pancreas is the acinus. We've met that term before. An acinus, you'll remember, is a ball of cells that has a lumen in the middle. All the cells in that ball are going to secrete their product into the lumen. The lumen will lead into a duct to take that secretion off to wherever it's going to go. So the pancreas is easy to confuse with salivary glands because they look very, very similar. I will tell you how to differentiate them in lab. So each acinus is made up of about 40 to 50 cells. And in the middle of the acinus, there are some special cells which are called centroacinar cells. If you look at this picture right here, you can see these cells here. Those are centroacinar cells. Now, I am sure that centroacinar cells were named before people knew what they were, because really what they are is part of the duct system. They're just part of the walls of the duct. But probably when people first discovered it, they said, oh, there's a cell in the middle of this acinus, so let's call it a centroacinar cell. Okay, so they are the beginning of the duct system. You can see that here quite clearly. And here's another picture showing the same thing. And here's a higher magnification. So these are the centroacinar cells. Notice they're just part of this duct, is all they are. So the shape of the cell is quite different from the shape of the acinar secretory cell. All right, acinar cells are pyramidal in shape. They sit on the basal lamina. They have a basally located nucleus that is typically spherical. The cytoplasm is basophilic. The apical part of the cell is filled with secretory granules. These are protein-producing cells. They're making enzymes, digestive enzymes. So they're going to have the features of a protein-producing cell. Euchromatic nucleus, prominent nucleolus, lots of FER, prominent Golgi apparatus, secretory vesicles, and the apex of the cell. The basal membrane has receptors for cholecystokinin. Now, have you already studied cholecystokinin in your other courses? So what does the name cholecystokinin imply? Chole means what? It means bile. Cyst means bladder. So what is the bile bladder? What organ is that? Gallbladder. Gall and bile mean the same thing. So gall is an older term for bile. Gallbladder. Kinin means what? Kinesis? What's the word I'm looking for? It means activity, or function. So cholecystokinin was named that because it activates or acts upon the gallbladder and causes the gallbladder to release bile for digestion. So that's where the name of the hormone comes from. Now obviously, if these cells have receptors for cholecystokinin, then cholecystokinin does more than just activate the gallbladder. It obviously has to activate these cells as well. There's a lot of rough-ER, polysomes, mitochondria, well-developed Golgi apparatus, and so forth. Okay, so they are stimulated by a hormone called secretin to produce a fluid that's high in bicarbonate ion. Why do you think it's high in bicarbonate? Yes, because it's going to secrete into the duodenum, and remember the duodenum just received this acidic junk from the stomach. And so this is going to help to neutralize that acid coming from the stomach. It's stimulated by cholecystokinin to secrete its proenzymes. You know what a proenzyme is, right? It's going to be cleaved to make an active enzyme later on. Parasympathetic innervation stimulates the acinar cells and the centroacinar cells as well. So there's several different factors that stimulate secretion. There are hormones, and there is parasympathetic innervation. What are the enzymes? We have pancreatic amylase, which breaks down carbohydrates. We have pancreatic lipase, which breaks down lipids, obviously. We have ribonuclease and DNase that break down nucleic acids. We have trypsinogen, chymotrypsinogen, that break down proteins. We have procarboxypeptidase, I can't even say it, elastase. So the names of the enzymes tell you sort of the substrates that they work on. All right. Now here is a human pancreas, and so you can see that it's made up of these little balls of cells called acini. Here's a higher magnification view. So here's an acinus right here. You can see some cells in the center, so those would be the centroacinar cells. Those are the ones. Here's another real prominent centroacinar cell. So those are the cells that form the first part of the duct system. Right there is one. Here are a couple of centroacinar cells and so forth. So this is what human pancreas looks like. Here is a picture of monkey pancreas, and the only reason why I put this in is because it shows the cells a little more clearly than the other one does. You can see the acini quite nicely, and the nuclei are located peripherally, and the apex is filled with these secretion granules. So this is a nice example of what pancreas looks like. Human and monkey are very similar. This picture is a little nicer. I think the reason why animal tissues come out nicer than human tissues sometimes is that when we prepare the tissues of animals, we're able to perfusion fix the animals. What we do is we actually run a line into the heart of the animal with fixative in it and run that fixative through the blood stream before the animal's actually dead. We anesthetize the animal so the animal feels no pain, and then we run this fixative through so the cells are fixed before they have a chance to necrose. Whereas with human tissue, we can't perfusion fix the tissue, so we have to wait till the person is dead and then harvest the tissue. So often animal tissues come out a little nicer in histological sections than human. There, what is that cell that I'm pointing at? It's a centroacinar cell. It's located right in the middle of the acinus. Okay. Very good. All right. So the centroacinar cells are squamous or locuboidal in shape. They have receptors for secretin, so they are also activated by hormones. And they form the intra-acinar portion of what we call intercalated ducts. Now, you've heard the word intercalated. Where have we encountered that word before? Cardiac muscle. So there's intercalated discs between the muscle fibers. So intercalated means located between other structures. They're called intercalated discs because they run between the acini. But they originate inside of the acini, and that's what the centroacinar cells are. Okay, so the intercalated ducts are the smallest ducts. Those are the ones that begin in the acini, and they're named because of their location. And then those are going to flow into larger ducts, which we call intralateral lobular. Remember, a lobule is defined by connective tissue septa. All the cells between the septa form one lobule. So there's ducts in the lobules, which we call intralobular ducts. And those flow into interlobular ducts. These are ducts that are going to be embedded in connective tissue between the lobules. And then those flow into the main pancreatic duct, which joins the common bile duct coming from the liver to secrete its product into the second part of the duodenum, the descending part of the duodenum. Okay? So there you go. So here is a nice, this is probably monkey again, I'm guessing, I don't know. Here's a nice acinus here. Here are the centroacinar cells, which are the beginning of the intercalated duct. And so you can see this duct is running between acini, so that's why we call it intercalated. It's intercalated between the acini. Here's a diagram of the same thing. Sometimes you can see a little clearer in a drawing than you can in a photograph. So this is just showing the intercalated duct. And notice that these intercalated ducts then are going to flow into an intralobular duct. And then that will flow to an interlobular duct and then into the main duct. All right. So that's the exocrine part of the pancreas. The endocrine part is more simple and yet more complex at the same time. Simple because the endocrine part is just these little pockets of endocrine cells that are easy to pick out, they're easy to see, but their function is quite complex. So let's talk about the endocrine portion. The endocrine portion is made up of pancreatic islets. Now, as I've said many times, many anatomical and histological structures were named after people. And we named them that to honor the person who discovered them, and that's fine, but Langerhans, who's Langerhans? We don't know who he is. So rather than saying islet of Langerhans, we now say pancreatic islet. Now most physicians probably still use islets of Langerhans, and you'll hear people say that. So you need to sort of know both terms. What does the word islet mean? A little island. A little island. So they're little islands of cells surrounded by the exocrine pancreas. Okay? There's about 1 million of these per pancreas, and obviously that varies from person to person. They're richly vascularized because they're producing hormones, and they have to release their hormones into the vascular system. So each islet is comprised of roughly 3,000 cells. I'm sure that some are bigger than others, and of course when you look at a tissue section, you may have cut through the center of an islet, which will make it look very big, or you may have cut through the periphery of an islet, which you only see a little piece of it. So you can't go by the size. They're most abundant in the tail region of the pancreas, and they're surrounded by and pervaded by reticular fibers. What are reticular fibers made of? Type 4 collagen, is he right? Type 3 collagen, are you guessing? You say 3? I say 3. And so I'm the boss, so it's 3. No, I'm kidding. Yeah, type 3 collagen. Type 4 collagen is basal laminar. Type 3 collagen is reticular fibers. Okay, there's multiple types of cells found in the pancreatic islets. The most prevalent type, the most abundant type, is the B cell or beta cell, and this is the one that produces insulin. You're all familiar with insulin. Insulin is necessary for the utilization of glucose by our cells. So people who don't have insulin are diabetic. Okay? So the second set of cells is called alpha cells or A cells. They are the second most abundant type. And what you're seeing in this picture and in the previous one is a tissue stained by a process which we call immunohistochemistry. What we do is we raise an antibody against that specific protein. We incubate the tissue with the antibody, and so anywhere in that tissue where that protein is located, the antibody is going to bind to it. Then we bind a secondary antibody to the primary one that has a color molecule on it so that we can actually see color at the places where that protein is located. So any cell in this particular thing that turns brown is going to be an A cell or an alpha cell. So those are the cells that produce glucagon. We'll talk about the function of these hormones in just a moment. You've already gone over it in physiology, so you probably could teach me. Delta cells produce somatostatin. The word somatostatin means, soma is what? Body. Statin, to stop. So it has to do with growth, or stopping growth is what it literally means. G cells produce gastrin. The G is for gastrin. And gastrin is a hormone that activates stomach function, but it also affects other parts of the digestive tract. There's PP cells, also called F cells, that produce a hormone called pancreatic polypeptide. Has some alliteration, so you can see there's not very many of those. And they tend to be at the periphery, which is another P. All right, so let's go through the hormones and talk about their function now. Insulin is produced as proinsulin in rough endoplasmic reticulum, is converted to proinsulin in rough endoplasmic cisternae, and then converted to active insulin in the Golgi apparatus where it's packaged for export. It is released in response to an increase in blood glucose levels. So its role is to decrease blood glucose by allowing cells to take up and utilize the glucose. All right, it increases cellular permeability to glucose. The second hormone is glucagon, and it is sort of the antagonist to insulin, so it does kind of the opposite thing. It is released in response to low glucose levels. It produces a prohormone, which undergoes proteolytic cleavage, acts on hepatocytes, that is liver cells, activating the glycogenolysis pathway, and it also, I don't know what it has to do with hydrolytic enzymes released in the bloodstream, whatever that means, and it activates gluconeogenesis. Okay, so its role is to increase blood glucose if there's not enough glucose. So if you have glycogen, it'll break glucose down and release glucose. If you don't have enough glycogen, you'll develop glucose from other things. You know about the gluconeogenesis, right? You studied that. Okay. Somatostatin inhibits the release of hormones by the alpha and beta cells. It suppresses the exocrine secretion of the pancreas. So it sort of goes against the digestive process. Reduces motility of smooth muscle in the digestive tract and gallbladder. So in other words, when digestion is over, this guy kicks in and sort of brings digestion to a halt. It's released due to increases in blood glucose, amino acids, or chylomicrons. Then we have gastrin. It's released by G cells, stimulates gastric release of hydrochloric acid, so it's going to act on the... What cells produce hydrochloric acid? Parietal cells, good. It stimulates gastric motility, gastric emptying. It stimulates regenerative cells to divide. And then we have pancreatic polypeptide, which is released by the PP cells and help to regulate both endocrine and exocrine functions of the pancreas. Okay, so that was a whirlwind through the pancreas. I think you probably already studied pancreatic enzymes and pancreatic function in physiology. Is that correct? Dr. Teal? Not yet? You will. Okay, you will. Okay, so when you get to Dr. Teal, remember where all these things are coming from, where they're being secreted, and then he'll give you more precise information about their function. We hope. Okay, liver. Liver is required to live. Maybe that's why they call it liver. No, liver, I don't know where the word liver comes from. I have no idea. Weighs about 1.5 kilos, so it's one of the larger organs in the body. Located in the upper right quadrant of the abdominal cavity, so just under the rib cage on the right side. Now, physicians will sometimes palpate to see if they can feel the liver, and if they can feel, that helps them to diagnose certain problems. I don't know what those problems are. Liver, like pancreas, has both endocrine and exocrine function. We typically think of liver as an exocrine organ, which it is, but it also has some endocrine function, which we'll talk about. One of the important functions of liver is in breaking down chemicals, drugs, toxins, and so forth. This is the reason why if you take too many of certain types of medications, they can overwhelm and destroy liver tissue. And an example of that's Tylenol. If you take Tylenol once in a while, it's fine. But if you take a lot of Tylenol all the time, it can destroy your liver. Alcohol, as you know, consuming alcohol is very dangerous in large quantities. And one of the common problems is cirrhosis, which is destruction of the liver. Well, if the liver doesn't work, you don't work. Okay? So, what is the structure of the liver? It's covered by a connective tissue capsule. This capsule happens to have a name. It's called Glisson's capsule, for whatever reason. The hilum of the liver is called the porta hepatis. Remember with Dr. Escobar, you talked about the portal vein. Okay? One of the reasons why that vein is called the portal vein is because it's entering the liver at the porta hepatis. So, porta means doorway or entryway. Porta hepatis, doorway to the liver. That's what the name means. Sort of a poetic name. So it's the entrance of blood vessels, nerves, lymphatics, and it's the exit of bile ducts, because bile is coming from the liver. Everything else is going to the liver. Okay? The main cell type in the liver is called the hepatocyte. Now, I was in a play many years ago in Redlands, in a little theater called Lifehouse Theater, and embarrassingly enough, the play was Heidi. The little, I don't know if you know that story, it's an old story about a little Swiss girl who lives in the Alps and lives with her grandfather. And Heidi's best friend was this crippled girl named Clara, and I played Clara's father, Herr Sesemann. Okay, so anyway, why am I telling you the story? Because we needed a kitten for the play, a live kitten. And so I went to the animal shelter, and I got this little black and white tuxedo cat, and took the kitten to the play, and then, of course, the audience went crazy. You know, we take the kitten out of the basket, everybody go, oh, you know, one of those things. And so we were making a cast t-shirt, so that all the cast members' names went back, and they said, what's your cat's name? And I said, I haven't named it. And they said, well, just tell me the name. I said, hepatocyte. Only an anatomist can come up with that. So on my cast t-shirt it says hepatocyte. So hepatocytes are organized in lobules. Now, a lobule in the liver is a little different than lobules that we've seen in other organs, in other glandular organs, because remember, in things like the salivary glands and the pancreas, you have connective tissue septa extending into the organ and breaking it up into lobules. That is not true in the human liver. It's true in other species, but not in humans. So we identify lobules by finding blood vessels and looking at the location of the blood vessels, and those form the boundaries of lobules. I'll show you what I mean in just a second. So, there are three different ways of describing lobules in the liver. And I want you to understand each type of lobule and what it describes. And I will test you over this, so be prepared for this, okay? If you look at this picture, all of these little pink cells are hepatocytes. And notice that between the hepatocytes, you can see little white spaces. Those white spaces are called sinusoids. Those are blood spaces. All right, so blood is flowing through the sinusoids. Now, this liver obviously has been fixed and stained, so the sinusoids don't show blood anymore. But in a living person, they would have blood in them. Notice that there's this dotted line surrounding this group of hepatocytes, and this dotted line roughly outlines a hexagon. It's a weird hexagon, but it kind of looks like a hexagon. Notice that at each angle of the hexagon, there are vascular structures. Remember when Dr. Escobar talked to you about the portal triad? Remember that? What are the constituents of the portal triad? Portal vein, hepatic artery, and bile duct. Those three things. Now, those three things are either entering or exiting the liver at the porta hepatis, but they all stay together even at the microscopic level, so they have the same branching pattern. And they all stay together as a little clump of three things. And so here, what we're seeing is little portal triads made up of branches of the portal vein, branches of the hepatic artery, and tiny little bile ductules that are going in the opposite direction, so they're going to merge to form bigger ducts until you finally get out to the bile ducts outside the liver. Okay? So we would call these little miniature portal triads. So each one of these intersections is called a portal area. So we have a portal area, 6 of them for each lobule. And then notice in the middle of the lobule, there is a single vein. We call that the central vein. So the classic liver lobule is formed by a central vein, and then the outline of that lobule is made by connecting the dots between 6 portal areas to form a hexagon. All right, so the central vein forms the longitudinal axis. Now we're looking at a cross-section through this lobule, but the real lobule is like a hot dog. You know, it's cylindrical in shape. We're looking at a cross-section through the cylinder. All right? Portal areas containing the portal vein, a hepatic artery, and a bile ductule form the 6 angles of this hexagon. All right? Hepatocyte cords, these little clusters of cells, radiate out from the center. You can see that sort of radiating pattern here. And so what's going to happen, you're going to learn, is that blood from the vein and the artery are going to flow from the portal areas to the central vein. Okay, so all the blood is flowing toward the center of this lobule. Bile is not floating toward the central vein because bile is not in the bloodstream. It's a separate system. So we'll learn about bile in a minute. So this just describes the flow of blood right now. Okay, so sinusoids, these little vascular spaces, are found between the hepatocyte cords. And blood percolates from the portal areas to the central vein. So we have blood flowing from each of the portal areas toward the central vein. Now notice that each portal area is shared between 3 lobules. And so some of the blood is going to flow toward this central vein, some is going to flow toward this central vein, and some is going to flow toward this central vein. Because all the lobules are sharing boundaries with each other. Okay? So each portal area supplies 3 lobules. All right? The central veins are going to flow into what we call sublobular veins, which flow into collecting veins, which ultimately flow into the hepatic veins, which flow to the inferior vena cava. So in this diagram, this cartoon right here describes the classic liver lobule. Portal areas at all 6 corners and blood flowing toward the central vein. Okay? So the classic lobule describes blood flow in the liver as its function. Now let's go to this, that's the classic lobule there. Now let's go to the second type, and this is called the portal lobule. My little circle got off a little bit, and I can't handle that, so I'm going to move it. That's a little bit better anyway. Okay? And of course I have to do this again. All right, so there we go. So the portal lobule describes the pattern of bile flow to interlobular bile ducts. So notice what happens here. This is very different. So you see that there's a triangle. And what links the triangle? The triangle links 3 central veins. And notice that at the center of this lobule, you have a portal area. All right? So any hepatocytes within that triangle, the bile produced by those hepatocytes is going to flow into that portal area in the central of that triangle. Okay? So bile flows in the opposite direction of blood. Bile is flowing out of the liver, blood is flowing into the liver. So bile flows to the portal areas because that's where the bile ducts are located. And then those ducts are going to coalesce to form bigger ducts and bigger ducts until they lead out of the liver down to the duodenum. Okay? So, what are the boundaries? The boundaries are imaginary lines between 3 portal central veins, as you can see in this triangle here. And the arrows indicate the direction of bile flow. Okay, so the bile is flowing toward this bile duct right here in this portal area. So the periphery is an imaginary line that connects 3 central veins. Our third and final, and again my circle is off, but anyway, you know it, this triangle, this diamond shape is what I want, so forget the circle. All right, the last one is called a hepatic acinus. We know the word acinus, but we haven't seen it used in this context before. This one is the most difficult to conceptualize. So notice that there is no central structure here. There's not a central vein, there's not a portal area. But the boundaries are, there's a long axis and a short axis. And the long axis is limited by 2 central veins, and the short axis is limited by 2 portal areas. Now what this diagram doesn't show is that boundary between, right in the middle of that drawing, that boundary, there is a little artery called a distributing artery that is coming from the portal area. So it's a little branch of an artery going between adjacent classical lobules. And this one's a hard one to explain. All right, so it's based on the blood flow from distributing arteries. So what this doesn't show is there's a little artery right here, and it has a branch that's running right down here. The branch is not shown. I don't know why the artists didn't show it, but they didn't. But there's an artery running at the boundary between two of the classic lobules. And so the area inside of the diamond shape is the area that's going to be perfused by the blood from that particular little distributing artery. Now, let's say that that artery is carrying some toxins in it. Which cells within that diamond are going to be the most impacted by the toxins? The ones that are next to the artery or the ones that are farthest away from the artery? The ones that are near the artery. And the farther you get from the artery, the less impact those toxins will have. All right? What if the blood is hypoxic? That means that it has too low of an oxygen content. Now, which cells are going to get the most oxygen? The ones nearby or the ones far away? The ones closest to the artery are going to get most of the oxygen. And so the ones farthest away are going to be the most impacted by the hypoxia. So, if it's hypoxia, the cells closest to the central vein are going to be the ones most impacted. If it's toxins, the cells farthest from the central veins and nearest to the distributing artery are going to get the most impact. Does that make sense? So this acinus describes the areas most affected by things like hypoxias or toxins. You can predict which cells will be the most impacted, which cells will be the least impacted. So this one is a little complicated compared to the other two. It's not as straightforward. So it indicates the order in which hepatocytes will degenerate due to toxicity or hypoxia. Now, toxicity and hypoxia are going to have opposite effects. Toxicity is going to affect the cells closest to the artery. Hypoxia is going to affect the cells farthest from the artery. Does that make sense? Okay. And so, within that little diamond shape, we divide it into 3 zones. And so here are the 3 zones. Zone 1 is right around the little artery. And so if it's a question of hypoxia, these are the cells that are going to get the most oxygen. I'm sorry, my time's up. Let me just finish this. Zone 2 is going to be more impacted by hypoxia, and zone 3 is going to be the most impacted by hypoxia. If we're talking about toxins, zone 1 is going to be the most impacted, zone 2 less impacted, and zone 3 least impacted. So it depends what is happening with the blood, whether they're going to be impacted or not. Now, as you can see, if you just look at liver under the microscope, none of these globules are going to be outlined for you. So you have to superimpose in your own mind as you're looking at the tissue, the triangle or the diamond or the hexagon, depending on what you're talking about. In my quiz or in my exam question, I will say, when considering the effect of toxins on hepatocytes, which type of lobule do we refer to? And it would be the acinus. If I say, when you're considering the flow of bile in the liver, which type of lobule will you refer to? And that would be the, whatever it is. Okay? I will let you go because my time is up, and we'll try to